Hello, everyone, and welcome back to the 50th Annual AAKP National Patient Meeting. If you're just joining us here or online, my name is Edward Hickey, and I'm the president of the American Association of Kidney Patients, the chair of the Veterans Health Initiative, and a fellow chronic kidney patient. I also have experience as a care partner for people in my family with a history of kidney disease. Our theme for this year's milestone event, 50 proud years of patient education, 50 years of saving lives, 50 years of saving livelihoods, reflects both the extraordinary progress we've made and the enduring mission that guides us. For 5 decades, AAKP has been committed to empowering patients, supporting innovation, and driving policy change in kidney care. This meeting is not only a celebration of our past, but also a look forward to the future we are pursuing together, one of greater patient care choice, stronger communities, and better outcomes for all impacted by kidney disease. It's my privilege to introduce to you today's session, Precision Medicine and Nephrology, Tailoring Treatment for CKD, which will feature 3 outstanding expert speakers, including Dr. Robert Starr, Director of the Division of Kidney, Urologic, and Hematologic Disease at the National Institute of Diabetes and Digestive and Kidney Diseases, which is part of the National Institute of Health. Rob also leads NIDDK's Renal Diagnostics and Therapeutics unit, where his team studies sepsis and acute kidney injury with a focus on early detection and treatment strategies. Also joining us is Dr. Kumar Sharma. Dr. Sharma is the L. David Hillis Distinguished Chair in Medicine, Chief of Nephrology, Vice Chair of Research, and Founding Director of the Center for Precision Medicine at UT Health San Antonio. His groundbreaking research uses multi-omic approaches to help understand kidney disease and identify new biomarkers and therapies. And lastly, Dr. Anand Srivastava, who is an associate professor of medicine at the University of Illinois at Chicago. He's a clinician scientist focused on discovering and validating non-invasive biomarkers to identify high-risk kidney patients, improve kidney trials, and guide drug development. Although Dr. Starr wasn't able to join us in person, this topic is one of his true passions, and we're thrilled to share his insights with you. He kindly recorded his presentation in advance of the meeting. Please turn your attention to the screen as Dr. Starr provides an update on the NIH-supported Kidney Precision Medicine Program.
Ed, thank you. Thank you very much for the opportunity to talk about the Kidney Precision Medicine Project. I really wish I were there. I would love to feel the energy in the room. So I'm feeling it now, but not quite. The Kidney Precision Medicine Project we'll be talking about is what is it? Why do we need it? What great science is already catalyzed by it? And critically, what still is needed? And I really love talking to the AAKP audiences because you get it. You've helped us in an uncountable number of ways, and we are happy to serve you because together, working together, we can advance innovations in kidney health. And I do have an assignment for you at the end, so please stay tuned. I always start with what I've learned by talking to people with kidney disease, like all the people in this audience, because you've told us what you want to know. And you want to know, what do I have? What will happen to me? What can I do about it? And what does it mean for my family? And that you much prefer a precision approach where you get the right intervention that's designed for you at the right time, a so-called precision medicine approach. What people with kidney disease do not want, we've also learned, is that now that you do have choices, and there are at least 4 classes of drugs out there, including the new SGLT2 inhibitors, GLP-1 agonists, that work in about a third to half of patients, that means that they don't work in the other half of patients. So patients don't want to be on an ineffective medicine for 2 years, because that's about how long it takes to figure out that it's not really working, and then try another drug for 2 years and see that it's not working. and then try another drug for 2 years, and then maybe get on the right drug. But this has been a waste of time and very dangerous because you've accumulated kidney damage and damage to your heart and your cardiovascular system, or maybe ended up on dialysis. And in the case of acute kidney injury, you really only get one chance. Instead, what we want to develop is a precision medicine or precision health approach. So shown here are 4 people with 4 different kinds of chronic kidney disease, CKD, plural. The idea is to have them undergo a series of fairly simple lab tests to figure out what color of kidney disease do they have, blue, red, yellow, or black. And then match the patients up with the pills that have been designed for their particular color of kidney disease. That's the right therapy for the right patient at the right time, and we hope leading to treatment success instead of the treatment failure on the last slide. So what is KPMP? KPMP is now in its 18th year, and we hope to have funding for at least 15 years. It's a multi-year collaboration that studies patients with the most common causes of kidney disease, including acute kidney injury and chronic kidney disease from high blood pressure, hypertension, and diabetes. And the goal is to define those disease subgroups, those 4 different colors of kidney disease, and the pathways that are causing the disease in the cells that are causing the disease that allow targeting of novel therapies. As you'll hear on the next few slides, patient engagement from people like you is at the core of KPMP. Patients with kidney disease sit on all of the KPMP committees and all of the working groups, and they were there, I'd like to say, from day -1, before we even conceptualized what this project was going to be. The goal of KPMP is to understand human kidney disease to improve treatment. We seek to, as I've said, redefine and reclassify common kidney diseases by combining some pretty fancy molecular and histologic, pathologic techniques that I will explain in the next couple of slides, along with clinical outcomes. And it all starts with a kidney biopsy. KPMP prides itself in spending a lot of time understanding the ethics of doing a kidney biopsy and how to do it safely, and that is the core mission of KPMP, ethics and safety. The study populations who we use are patients with diabetes and hypertension, as well as acute kidney injury. We combine the clinical data with the sort of traditional pathology data, the very fancy new omics and imaging, which I'll show you, as well as clinical outcomes to identify the cells that are causing the disease, looking inside the cells to ask what pathways, what genetic mechanistic pathways have been activated in order to find targets for novel therapies and develop these mechanism-based disease subtypes and drugs, your name. Here, all this data goes into a repository called the Kidney Tissue Atlas that's available to the entire research community, academia, and pharma. And believe me, pharma is using this data every day. As of last month, there were 630 biopsies in KPMP, 3/4 in chronic kidney disease, 15% of the patients had acute kidney injury, and 10% were from reference populations, other people that we needed to compare against. People with kidney disease fit into all aspects of KPMP because we learned from Richard Knight from AAKP, don't do anything to me without me. That is a mantra of the design of KPMP. They're on all the committees. They wrote the initial informed consent document. They've helped with the website. They've helped with communication, community outreach. They make sure that the whole study stays on track and works towards developing new therapies for patients with kidney disease. As Paul Conway has said, KPMP is the global standard for patient engagement, and we intend to keep it that way. I'd like to begin to look under the hood of KPMP and show you what it's actually doing. To do that, I'd like to start with this slide that shows the 4 subgroups of patients with kidney disease, different colors. We've seen this before. Each of those pathways gets to their kidney disease and goes through it in a different pathway. Those are the 4 arrows, 4 different colored arrows to whatever outcome the individual patients get. There are 4 major clinical or scientific questions for precision medicine. The first one is, what biology do I have? What color kidney disease what's causing it, and what's a marker to show that I have that blue kidney disease. Second question is, what can I do about it? Okay, I have the blue kidney disease. What drug treats patients with the blue kidney disease? And is that drug working? And can you figure that out quickly in a month or two and not have to wait 1 or 2 years to see whether the drug is actually working? That's an early drug response marker. The fourth question is the biology or mediator, is the biomarkers, what group I'm in, the drug, number 2, and is the drug working, 3, on the same biological path and very, very close to each other on the same pathway. It's taken me 20 years to figure that close piece out. Notice that these questions are very similar to the 4 patient questions. What do I have? What's my biology? What can I do about it? What drug should I be on? What will happen to me? Is the drug working? And what about my family pathway? What we've learned in KPMP is that our current clinical and pathologic classification systems do not work very well. This slide on the left-hand slide shows the diagnoses that people came in at enrollment at the beginning of the study: hypertension or high blood pressure, diabetes, or acute kidney injury. And after the KPMP scientists have reviewed all the data, clinical data, including molecular data that they have, we discovered that many patients need to be reclassified into a new diagnosis, and all those squiggly lines show the reclassifications that are going on. The major ones are shown here. A lot of the clinical type people thought to be diabetic chronic kidney disease actually have hypertensive kidney disease. The hypertensive kidney disease bucket is actually a huge mix. And many people who we didn't suspect had acute kidney injury, which is a fascinating concept and a whole talk in and of itself. There's a quote that's attributed to Albert Einstein, but it's more accurately probably attributed to a mystery novelist, Rita Mae Brown, and that is, insanity is doing the same thing over and over and over again and expecting a different outcome. And that's where the kidney community has been for the last 75 years until very recently. Instead, the call to action to KPMP and with KPMP is to leverage all of our expertise from academia, from pharma, from industry, and all of our data, including KPMP and beyond, in order to discover and define the new disease pathways, those 4 colored pathways I talked about, the 4 subgroups, 4 colored patients I showed you, that enable precision clinical trials. KPMP has catalyzed an explosion of new science, which I'll be talking about over the next couple of slides. And the KPMP data is being used incredibly widely. Many, many talks at the American Society of Nephrology meeting mentioned KPMP, and pharma we know is downloading the data and using it to check their drug targets. To open the hood a little bit, I'd like to take you through a couple pathways of discovery. So what KPMP is looking for is what causes injury, what could increase repair of injured cells, or is there a way to make cells or the kidney more resilient to injury, and then look for potential drug targets for these kinds of questions. There are 2 principal ways to do this. The first path is a pathology path that involves looking at tissue sections, at the biopsies themselves, looking for patterns of injury, new measures, and neighborhood hotspots. I will show you these 3 things on the next couple of slides. The second pathway is a biology pathway, where again you're looking at neighborhood hotspots, but looking at the cells of injury, looking inside the cells, what pathways have been activated, and looking for specific mechanisms of either injury repair or resilience, using both the pathology and biology pathways to find potential drug targets, which then give us some idea of how you might define the subgroups that would be attached to those potential drug targets, and then bring that back to the beginning. It turns out that we have to go around this circle a couple of times. Each time we refine the subgroups, the biology, the pathology, the targets, to get to our long-term goal of individualized patient health, that drug that has your name on it. So we're now going to go through a bunch of somewhat complicated slides, and on almost all of them in yellow, I have put the punchline. So if you're having trouble following what's on the slide, just look at the punchline and listen to it say. So this slide shows sort of what you would get, 50 years ago from a biopsy. It's the histology of kidney injury, in this case, chronic kidney disease. The tubules at the top are in red. Some of them look pretty good, some of them not. The ones at the bottom look really bad. And all that blue stuff is scar tissue, fibrosis. So this is a pattern of injury. There's fibrosis. That tells you a little bit, but not enough to find good treatments. If you take 1,000 patient samples and scan them digitally, put them into a computer, and ask the computer, can you unravel what's going on? Can you find some new measures? The computer has done this using machine learning. They found 4 measures, some of them new, some old, 2 from tubules, 3 from glomeruli. Tubular distance, the distance between the tubules, how wide are the tubules, what do the glomerular tufts look like, are they circular, are they eccentric? If you take all the glomeruli that they looked at from those 1,000 patient samples and plot them using fancy mathematics, you get a pseudotimes arrangement. The color dots show the degree of injury from little injury black to a lot of injury yellow, and the black line shows how the disease is progressing through a disease pseudotime. This isn't any individual patient, it's no individual glomeruli. But when you ask, okay, give me representative glomeruli of these different stages, what you see is now at the top. And it shows on the left a normal-looking glomeruli and how a glomerulus might undergo damage until all the way at the end it looks really bad. What has happened here is the mathematics have stitched together disease progression from individual glomeruli in individual patients to unravel this very complicated disease trajectory. Now that's nice, but let's push it a little more. When you now look at the individual measures and see, for example, if the tubules are packed in close to each other in blue, the patients do pretty well. This is a survival curve. It sort of shows progression of kidney disease. And if the tubules are wider apart, the progression is much faster. Same for tough cystometry. And why is this important? Okay, punchline. This digital pathology, unraveled by machine learning, not by people, can predict more accurately who has worse prognosis. It's not giving us drug targets, but it's giving us an idea of where to go look for disease and how to grade it, which turns out to be really important. Now let's push this some more. In real estate, you know that location is important. Every structure, every house has an address in the city. Context matter. Where you live, what neighborhood you live in shapes the livability of the neighborhood and the health of its inhabitants. And maps show us how the businesses and houses create healthy neighborhoods or food deserts. Similarly, there are cellular neighborhoods in a biopsy sample. Every cell in there has an address, and the context matters. The cell neighbors shape its behavior, we're learning. The map shows us where the cells form healthy neighborhoods in green, or where they're diseased nephrons, the hotspots in red. Now, most of this we knew. The really exciting finding is on the next slide, where instead of looking at straight histology, we're now looking at genes. So this slide has been probed with a very expensive technique called spatial gene expression analysis, where you get gene signatures from each individual cell, and you know where the cells are because they're on the slide. And when you look at these gene signatures, you can see that they're different proximal tubule cell states, and they range all the way from healthy, shown in white, to severe injury, shown in green. And what's interesting is that the most severely injured ones, again, based on their gene expression, are living next to fibroblasts. Fibroblasts are the bad cells in the kidney that are creating that scar, that blue scar I showed you a couple slides before. So we can see how the neighbors influence where you are, which genes get expressed. So why is this important? We can now actually see and detect biological signaling between cells, and this is a way of finding new drug targets. To push this even more, and this slide's a little complicated, but basically we're looking at where you are in the nephron, the functional part of the kidney, combining that with whether the cell we're looking at is healthy, injured, getting worse, getting better, as well as the neighborhood where it's living based on its gene signature, kind of deduced from where it might be living, that the outcome actually changes, that in this case, activated thick ascending limb, different part of the kidney, but important for kidney health, that the activated thick ascending limb cells sitting in a bad neighborhood, those patients who have lots of them do worse. The green line is opposed to those patients that have fewer activated thick ascending limbs in the bad neighborhoods. Punchline. Biologic differences in cells make up the neighborhood, give the unique characteristics of the neighborhood, and the neighborhood alters the cells. This is important and clinically important because you can use this information to find out which patients are going to do worse and faster decline in kidney function. And it's a way of identifying different treatment options because we now know what the cells are and the activation state. We can tell what pathways have been activated. Okay, now we're going to push it even more. This is a map of the United States, of the highways in the United States. How do you get from point A to point B? There's probably 200 different ways. This could also be a map of the 20,000 genes in your body and how they are interconnected. And it's a real mess and difficult to unravel at this level. However, if you make a simplifying assumption and now simplify the map to the 100 or 200 functional gene modules, you're going to get a map that looks something like this, where it becomes a whole lot easier to see how we're going to get from point A to point B. There are only, maybe a handful of ways of getting there. Simplification helps us search for disease biology. And to show that, this is exactly what Dr. Sustak's lab did using KPMP data. She and her lab are not in KPMP, but they used our data, and they made some simplifying assumptions. And they basically found in patients with kidney disease, there was a overabundance of 3 pathways shown here. And what she then looked for was, where in those pathways are there not many roads? Where's their open road? Because that's where I might want to place a drug to block flow from A to B. So think of those paths as disease pathways that I showed you, those 3 different disease pathways I showed you. And it allows her to figure out a fairly complicated molecular scorecard profiling report where, based on the gene signatures in a kidney biopsy of an individual patient, she could classify which cells were causing problems and which pathways were potentially involved, and allowed her in a fairly simplified way in a small number of patients to test this out. So what she showed is that we actually can find a small number of gene modules, think of them as functional gene neighborhoods, that are dysregulated in an individual patient, and then suggest which ones to target where there may be no roads nearby that the drug might be interacting with. So this is a really important paper because it shows that the whole idea behind KPMP will work. We just need more data. But it needed to have the right simplification, and that was the beauty of this paper. This next paper is in a whole different category. So let me walk you through it. The left-hand side shows a fluorescent image of a kidney. The red balls are the glomeruli, and they're in a human kidney. There are, a million of them, and the tubules are shown in a lighter purple color. And we thought for the last, 200 years, that each nephron, each glomerulus acted on its own. It sensed what was going on, and it acted on its own, and it conserved salt and water by some local signaling within the individual nephron. That turns out not to be true. What we missed, and we knew there were renal nerves shown in green, but it took light sheet microscopy by a couple of KPMP investigators, although funded using a different NIH mechanism called HubMap, to realize that there actually is unsuspected communication between the nephrons carried on these renal nerves and their neighborhoods of glomeruli, some 2, some 5, some 7, 8, 9 clusters of glomeruli. And the important part is that they're all controlled by a mother glom. So the nephrons form different, distinct communities. These nephrons are linked via nerves, and this whole thing is controlled from a couple of mother glomeruli that sense what's going on and then send signals out to the rest of the glomeruli, telling them what to do. And these communities are disrupted in diabetes, Our challenge right now is to incorporate this new information that just came out into KPMP. And this shows the beauty of science, that we're always advancing. People are coming up using new methods to come up with really unbelievably new ideas that will help us understand how the kidney works and why it doesn't work, and find us a whole different set of drug targets that we never would have imagined. So our goal for the future is not just to understand kidney disease, but to improve treatment. And there are a few hurdles. How do we turn all this data into knowledge to actually help design new drugs? Some of this we understand how to do. The last couple of papers have shown at least a roadmap for how to get there. But the harder part is figuring out, how do we screen individual people to put them into the right trial? To do this one by one, each drug company has its own trial network and its own screening mechanism. Is very costly and inefficient and not good for patients, because you have to go to 5 of them individually. Why not have them come to you? So one way to gain efficiency in kidney disease clinical trials is to match patients to multiple drugs one time at once, using a common infrastructure. Have the infrastructure come to you, not you go to the infrastructure. So 37 million people with kidney disease get the blood tests that tell us which group they're in, what's their biology, what color kidney disease they have. They then get matched to a candidate therapy. This particular patient will be put into trial B, getting the red drug and leading hope. And then you could then test to see whether this precision medicine approach actually works. But let's do it efficiently. A bunch of things are needed, shown below, but one exciting prospect is that once you've been screened, even if you didn't match to any of the existing drugs, your blood is still available to test it against new drugs. Maybe you need the 5th or 6th drug that came out, not the first 4. So this seems to be a far more efficient way to do it, but it would require a real culture change in how clinical trials are run. And this is where we need your help. So my final thoughts are that our challenge here is that kidney disease does not fit in the existing box that we've placed it in. And the mother glomeruli slide really proves that point, that we need to reconceptualize kidney disease, how we treat kidney disease, and also kidney health. That's another whole talk. Please invite me back for that. As I've tried to show you, KPMP has a lot of beautiful new tools that it's catalyzed. There's new science out there, the mother glomeruli. We need more participants in KPMP. We're slowly getting them, and it needs to go longer. We're hoping to do that. But more critically, we need to tie the data together. The community needs to do this, not just the KPMP scientists, into actionable information that helps you. And then we need to design some kind of clinical trial network where it comes to the patient. And here's where we need your help to help us change the culture of how clinical trials are doing. The drug companies are beginning to listen to the patient voice. This would be good to talk to them about. So with that, I'd like to thank you for all your help in helping us advance innovations in kidney health, and look forward to continuing these dialogues with KPMP and its leadership over the next decade, because we need to keep moving forward. Thank you.
Thank you, Bob. We truly appreciate Dr. Sharp's remarks and the ongoing support of the NIH, NIDDK for the Kidney Precision Medicine Program. And as you'll hear from our next speakers, the patient data generated through KPMP is already providing important insights, helping us better understand kidney disease and driving future innovations. With that, I'd like to invite Dr. Kumar Sharma to the podium. Dr. Sharma.
Thank you, Ed, and it's a pleasure to be here, and the AAKP is doing so much for our patients. And I'd like to talk to you about how precision therapeutics could actually be here today based on some of the data that we're seeing and using a new approach that Dr. Starr did mention, which is called spatial metabolomics. So when we think about kidney disease, where we want to block kidney failure, we actually have a very good biomarker for that, and that's proteinuria. So many patients with chronic kidney disease get put into 2 buckets: those that have proteinuria and those that don't have proteinuria. Both groups can progress, but certainly the ones that have proteinuria progress a lot faster. And the more protein they have in their urine, the faster their kidney disease progresses. But there are a subset of patients that don't have proteinuria as well. And so what we want to do with the new technologies is to see how we can understand these two processes. And when we think about proteinuria, there's a cell type in the kidney that many of you may be familiar with. And if you're not, this is a very distinctive cell type called the podocytes. And we'll get into that. But when you have proteinuria, there is immediately a problem with your podocytes. And so we have to understand, why do podocytes fail in progressive kidney disease? The second major question that I think is involved in non-proteinuric kidney disease is inflammation and fibrosis. And you saw examples of that in the previous talk. But why do patients without proteinuria have inflammation and fibrosis? And how can we figure out what's driving it and potentially how to treat it? So here's an example of a problem with the podocytes. And in the top left panel, you see this barrier, which is essentially a wall to prevent protein to go from the blood side to the urine side. And the final warrior to prevent that protein to get across into the urine are these podocytes. And that's the cell type that you see there with these foot processes. However, when the podocytes lose their ability to stay on that near the wall and prevent the protein to get across, they can fall off. So we're not sure exactly why these podocytes fall off and lose their footing. But we do know with very large clinical studies that the higher the protein you have and the lower your GFR, the more likely you will have kidney failure. So if we can find out ways of blocking this progression of podocyte dropout, perhaps we can block proteinuria kidney disease and protect patients from developing kidney failure and needing dialysis. So the approach that we've been taking as part of KPMP and part of our Center for Precision Medicine is to use the metabolites to guide us to figure out what the potential therapeutic could be. And the way we do that is to work in a backward approach. So Dr. Starr talked to you about how to identify genes that may predict to go from A to B. But if we start at B and we think about what are the pathologies and the metabolites that are there and work backwards, perhaps we can short circuit that long journey to find a therapeutic. So we start with what's a technique called spatial metabolomics. And this is an example of what spatial metabolomics looks like. We get frozen kidney sections and we implement a matrix on it, shoot a laser on those frozen sections, and desorb the molecules that come from that section. And in this case, we found that with every single kidney that we looked at that was a normal kidney, that was a specific metabolite called sphingomyelin that was present in the glomeruli. And the podocytes are in the glomeruli. So we became very interested in trying to figure out, why is sphingomyelin in the glomeruli, and using a complicated bioinformatic approach with single-cell RNA-seq, we identified a gene called ceramide synthase 6 that was present in every single normal glomerulus. And this was confirmed by protein analysis as well as by gene analysis. And interestingly, when we looked at CER6 going across the stages from early diabetic kidney disease to mid-diabetic kidney disease to late diabetic kidney disease, we see that CER6 actually increases during the early and the middle stages of diabetic kidney disease, but is almost gone at the end of diabetic kidney disease. And these are the groups of patients that we had. So the patients had relatively normal GFR in the beginning with very little proteinuria. Oops, we lost our slide. There we go. Okay, great. And in the right panel, you could see that this amount of CER6 protein goes up in the glomeruli in early and middle stage diabetic kidney disease, but it's almost gone in late stage diabetic kidney disease. So what is this protein doing in the podocytes? And the way to figure that out is to do a study by removing the protein from those kidneys, and we did that with a knockout mouse. And what we found was very interesting, that when you remove this protein from the podocytes, that the kidneys are actually protected. So if you don't have this protein, you have less albuminuria, less scarring in the kidney, and improved kidney function with just aging. So the function of this protein seems to be important in a complex way. And the reason why it is having this complex pattern is because this CER6 protein produces a toxic metabolite called ceramide, C16 ceramide. And of all the ceramides that are made, which is a type of fatty acid, the C16 ceramide is particularly toxic to the podocyte. And so when the kidney is making too much of the C16 ceramide, the podocytes can't handle it, and then they drop out. So this is actually a therapeutic target. So now we have a new therapeutic target, and if we can block the C16 ceramide production, we can protect those podocytes and hopefully prevent proteinuria kidney disease. What about the patients who don't have proteinuria kidney disease and still have progression? This is actually 90% of diabetic patients, and many patients with hypertension also have non-proteinuric progressive kidney disease. If we do a biopsy and look at a frozen section, this is what the kidney would look like. If you have hypertension and kidney disease without proteinuria, there might be atrophic tubules, fibrosis, and vascular arteriosclerosis. But why does it come into the kidney? How does it get there? And again, the reason may be by figuring out which molecules are causing this problem in these neighborhoods. And what we found with spatial metabolomics is that a specific molecule called adenine is actually localized in the same region as the fibrotic areas and the sclerotic blood vessels. So we asked the question, could endogenous adenine be playing a role in fibrotic kidney disease? And the answer seems to be yes. We actually have a non-invasive biomarker called that we can measure adenine in the urine, and we find that those patients who have high urine adenine have a much higher rate of progression of kidney failure and mortality. And we found that in the US population as well as the population in Singapore. So across ethnicities, this increase in urine adenine seems to be a good marker for identifying patients with progressive kidney failure and mortality. But is it really toxic? Is it causing the problem? And the answer seems to be yes. If we block adenine production by blocking an enzyme that is responsible for making adenine, called MTAP, mice that have diabetic kidney disease are essentially protected, and they don't have progressive kidney disease if the adenine accumulation is blocked. And this is true not only for chronic kidney disease, but also for acute kidney injury. With ischemic kidney disease, MTAP inhibition to block adenine accumulation completely protects the kidney from ischemia and also from cisplatin, which is also very toxic to the kidney and used as an oncologic therapeutic agent. So both for chronic and acute kidney disease, endogenous adenine production seems to be very important. And not only is this true for the kidney, but also for the heart. So many of our patients with kidney disease get heart disease, but we find that blocking adenine can also be very protective for the heart in terms of heart infarcts. So our overall theory is that in many instances where you have some injury to the cells, adenine is released by the cell and causes damage. And this increase in adenine production causes cellular damage throughout that neighborhood and could be the reason why diseases progress both in the kidney and in the heart. So all of this data is very interesting and very speculative, but what can we do right now for our patients? So I want to tell you about a case that happened very recently in our hospital, in our laboratory. A student came to me and worked with me in the lab and said, I want to understand my own kidney disease. So the student has a condition called C3 glomerulopathy, which was diagnosed about 2 years ago, and his kidneys failed. He then underwent a kidney transplant, and this kidney that he got from his father also has C3 glomerulopathy. So he wanted to find out, why is his kidneys failing? And we were able to get the biopsy from 2 years ago because we developed a new method where we can now analyze not only frozen sections, but also something called FFPE blocks, which are stored for decades. And so anybody with a kidney disease can have that block section and analyzed by this method. And using that block, which he was able to get, we found that these metabolites were very highly present in the diseased portions of his glomeruli. And that included that molecule I talked about, adenine, another molecule called guanine, and a third molecule, thymine, which is in the pyrimidine pathway, was actually reduced. And so when we look at the ratio of purines and pyrimidines in the diseased glomeruli versus the healthy glomeruli, we find a marked increase in the purine-pyrimidine ratio. And why is that important? It's important because this increase in purine-pyrimidine ratio is known to be very toxic to cells generally. And many genetic diseases have an increase in purine-pyrimidine excess. And there's actually a way to treat it by blocking purine synthesis with antimetabolites such as allopurinol or azathioprine. So we now have a new way of treating this patient with progressive kidney disease based on his own kidney biopsy and the metabolites that we found there. And this treatment would not have been suggested with any other approach. So I think we're in a very exciting phase in a new era of precision medicine that's actually available right now. With spatial metabolomics, we can identify new precision biomarkers based on pathology features and match the treatment with these risk markers. There are new treatments that we can start developing that block C16 ceramide to protect against proteinuria kidney disease and to block adenine production that blocks inflammation and fibrosis in chronic kidney disease. We also can use these metabolite biomarkers not only for therapeutics, but also to guide dietary interventions. And finally, we're now entering this new era of innovative clinical trials where we can use these multiple biomarkers to find the right treatment for the right patient in both acute and chronic complex diseases. So thank you very much, and I'd be happy to answer any questions if we have time later on.
Thank you very much, Dr. Sharma. That was fascinating, and thank you for advancing the future of precision medicine and nephrology. Now it's my pleasure to welcome our final speaker, Dr. Anand Srivastava, who will share insights into his work leveraging findings from the Kidney Precision Medicine Program.
Thank you, Ed. Really appreciate it. And thank you to the organizers for the invitation to speak here today. Okay, great. So the title of this talk is actually Leveraging Functional MRI Biomarkers in Chronic Kidney Disease. Some of my disclosures, none of which are pertinent for the purpose of this talk. Only disclosure is I recognize that I am the final speaker before lunch, and so I will try to keep you engaged. All right. So this has already been relatively well described and is no secret to this audience, but Chronic kidney disease is bad. It affects over 30 million adults in the United States. It increases the risk for end-stage kidney disease, or ESKD as I'll refer to it, which is the need for dialysis or kidney transplantation. This is already old data, but the cost of treating this already approaches over $120 billion per year. And this is our CKD heat map, which has already been shown in Dr. Sharma's slides very nicely. But two major biomarkers, estimated glomerular filtration rate, or eGFR, and albuminuria, are two simple biomarkers, one in the blood and one in the urine, to estimate an individual's risk of this progressing. But the unfortunate part about eGFR and UACR, or albuminuria, is that they cannot really tell us about the underlying biology of what's going on in the kidney. They can help us assess risk, but they can't tell us what's going on overall with regards to if you want to develop a new therapy. So one potential pathway is about hypoxia or decreased oxygenation. And this is the chronic hypoxia hypothesis. The idea that after an individual develops kidney injury on the top right, you get worsening microvascular insufficiency that can lead to worse oxygenation or hypoxia. And that can lead to tubular injury, inflammation, and subsequently fibrosis that leads to capillary dropout. And this is a vicious cycle, as you can see. And that leads to progressive kidney disease, which is what you see as a decrease in estimated glomerular filtration rate. And actually, over time, the kidneys atrophy in size. So can you harness imaging in the blue at different phases of this pathway to be able to give you information about the kidney in real time that we can use in clinical trials and assess response to therapy? This is data that we produce in a biopsy series of almost 700 individuals to demonstrate the importance of these lesions in the kidney. So we looked and graded individuals with global glomerular sclerosis. So the glomerulus is the filtering apparatus of the kidney. And when it scars down, it leads to global glomerular sclerosis. Fibrosis, which Dr. Sharma nicely alluded to, arterial and arteriolar sclerosis. And what this shows is almost in a stepwise relationship fashion that with worsening grades of these lesions, you have an independently increased risk of the development of end-stage kidney disease or a drop in your GFR by greater than 40%. But this is where we are today. Still, 50 years later, we're still using this modality for imaging, which is typical grayscale B-mode ultrasound. Patients in the room can attest to they've had this done before. I would imagine that the majority of you have had something like this done before. The problem with this is that despite other fields of medicine advancing dramatically with non-invasive imaging and transforming their fields, our field has lagged behind it. And we still use this to this day. But it doesn't tell us very much information other than the size of the kidney about what's going on inside of it. So I'll give you a little bit of the objectives today is to give you some overview of functional magnetic resonance imaging, or fMRI as I'll call it, how we're using it in single and multi-center studies, and potentially the application of using it in assessing response to therapy in clinical trials. Just a bit of a primer of three different sequences that can be done in fMRI. So on the top left, we call it arterial spin labeling, or ASL. It gives us an assessment of perfusion in the kidney. So it doesn't use gadolinium like an injectable tracer. Actually, it uses your own blood and water, and it does a subtraction of the two and creates a perfusion map, and we can estimate the perfusion of the kidney in such way. On the bottom, you see blood oxygen level dependent, or BOLD MRI. It uses the paramagnetic properties of what we call deoxyhemoglobin and estimates the oxygenation in the kidney. And then diffusion-weighted MRI assesses the movement of water. And so an individual that has less water in their kidney may have a stiffer kidney, more fibrosis. These give you an assessment of fibrosis in the kidney, or at least we hope so. Of the biomarkers that are generated, I already mentioned ASL provides perfusion, BOLD provides an oxygenation, and diffusion-weighted MRI provides you a readout of fibrosis. On the right of the slides, you'll see just a typical healthy volunteer and then a person with chronic kidney disease. Visually, you can tell that these look different. These kidneys look different to you. But again, visually is great, but does it give you a quantitative measure that you can use and apply and differentiate people in that fashion? Part of the issue is, okay, this is great. We have these biomarkers, we can measure them. Is it actually measuring what we think we're measuring? This is a study that was done that shows when we injected a tracer into individuals, that's kind of the gold standard for assessment of kidney blood flow. Does the perfusion metric in the kidney from MRI give you an assessment of kidney blood flow? And the short answer is, we think so. It has probably a moderate to strong correlation with kidney blood flow and measured in such a fashion. You might say, why don't you just use that tracer in the clinic? The truth is doing that tracer in the clinic is about a 3 to 5 hour study and not something that you can do in relatively simple clinical practice. So the MRI might give you an estimate of this instead. How about oxygenation? Measuring oxygenation in the kidney is actually very challenging. You can really tough in animal studies, in fact. So this was a study that was done that looked at individuals, and they measured GFR in each of them in a very detailed fashion. They did MRIs, and they also did an angiogram of the kidney. And going from left to right, you see worsening in GFR, you see worse levels of oxygenation, and angiographically, you can see that there's less vasculature in those individuals with worse GFR. So it seems to be going in the direction that we think, that worse oxygenation, less vessels that you can use and recruit in individuals with worse chronic kidney disease. How about the fibrosis metric? Does it tell you about fibrosis in the kidney? This is data from almost 140 individuals that underwent a kidney biopsy for various reasons and showed that in individuals that had this done, that the correlation coefficient between the fibrosis measured from MRI and fibrosis that was measured in the kidney had a moderate to strong correlation overall. So overall, we think that these metrics are telling us about biologic and physiologic metrics in the kidney that may be pertinent. How about in single center studies? Can you differentiate people that may actually progress or do worse? This is, again, a study of some of our collaborators that looked at the perfusion sequence in MRI, and they followed people over time, and they saw that individuals who had lower kidney blood flow as assessed by MRI, those are the ones in red, they were more likely to be progressors and have worse kidney function over time. And the ones in green are the people who had better blood flow, and they were non-progressors. So you may be able to differentiate these types of individuals. This was a study that was highly cited, got a lot of press, and that individuals that had worse kidney oxygenation, you can see on the right, had an increased risk of kidney function decline over time. That's usually the line there on the bottom, showing that those individuals are more likely to reach the composite endpoint. So some of the work that we've done is incorporating this into a clinical trial, and this is one of the first multi-center studies to actually do this. Could we actually incorporate MRI into a clinical trial and be a reliable endpoint, and could it tell you about kidney health overall? So this is embedded into a trial of 205 individuals. We did the MRIs in almost 120 individuals and repeated it in about a subset of 90 of them. What it showed is that in a multi-center fashion, the biomarkers are reproducible. So each of those on the x-axis are the sites, 6 different sites, and then the biomarker measurements on the y-axis. And it shows relatively similar values, which it should be in a clinical trial of a relatively similar population. You should have biomarkers that are relatively similar. When we looked at change in GFR over time, we saw that the marker for fibrosis actually was associated with change in kidney function over time, meaning that worse fibrosis on the MRI associated with worse GFR decline over time. But after we adjusted further, very rigorously for changes in proteinuria over time, we saw that signal slightly go away, that other factors may explain why people's GFR changes over time. And while this is disappointing, it could actually be that these individuals had a very low GFR already, and we didn't have a good enough spectrum of individuals. We didn't have a large enough sample size, and there actually wasn't enough time in the trial because it was only over 1 year. What we did find is that in individuals that had a repeat, that over 1 year, these values were very stable. So there wasn't that much change over 1 year, which suggests that if you're going to embed it into a clinical trial, we probably should space out the measurements so that there's more time for GFR to change and more time for disease to set in. And that's where a lot of the work is going for us now in the Kidney Precision Medicine Project. Dr. Starr already basically explained this exact slide, but we are embedding this MRI technique into the Kidney Precision Medicine Project. So our goal that's funded by the NIDDK is to do this in 420 individuals across 8 clinical physical sites. And we'll test the associations of the biomarkers with the underlying lesions in the kidney. It's been done, as I showed you, in smaller sample sizes, but we really need to delve down deep and see, are we measuring what we think we're measuring in the kidney? And then we'll investigate these biomarkers with changes in kidney function over time. All of these people, as Dr. Starr mentioned, are followed for, we hope, 10 years, and we'll have a much better sample as to what's going on with these biomarkers and how they affect kidney function over time. And then we're going to use more sophisticated artificial intelligence techniques where we extract more features from the images themselves, not just the single biomarkers, but can you learn about the image themselves and the kidney and extract more features and almost develop different imaging subphenotypes of these individuals. And these are the recruitment sites that we're doing it in. So we will do it across the United States in these 8 physical sites. We'll do the MRI within about 4 to 6 weeks of a kidney biopsy that they're undergoing for research purposes and chronic kidney disease, and then we'll repeat the MRI 2 years later in a subset of the individuals. These are some of the examples of the data that we've already generated. These are 2 different individuals. Both had a kidney biopsy. One has a GFR of 55, the other one of over 90. One has UACR, this is the one on the left, albuminuria of 2 grams, and the one on the right, 1 gram. Their profiles of fibrosis and glomerulosclerosis and microvascular disease are relatively similar. But when you look at each of their imaging biomarkers, they actually are different. And you can see differences in their bold MRI values, differences in their diffusion-weighted MRI values, and in their perfusion values, all to suggest that while people can look somewhat similar histopathologically, different with clinical variables, they can still look quite different with imaging variables. So these tell you maybe a little bit more about their overall kidney health that we need to delve down deeper into. We've done this now, actually we've done it in about almost 100 individuals in the Kidney Precision Medicine Project. These are some data from the 83 people. These are people that we see in clinic. These are very similar to people I see almost every week in clinic. They're around 60 years of age, half of them have diabetes, GFR is around 60, albuminuria around 200 milligrams per gram. But interestingly, that even though these people's GFRs are relatively preserved in our clinics, you can see, and one of the findings from this project is that these individuals already have, many of them, a quarter to a third of them, already have pretty severe amounts of fibrosis in their kidney, despite having relatively preserved GFR. We've really delved down deep with these fibrosis measures and the imaging biomarkers to see if we're measuring it accurately. And we've sent it to 2 different image analysts, and we saw that these measurements are actually reproducible by 2 different readers read at different times who are agnostic to each other. We also saw that when this was done, the measurements they actually got were within a similar range. But these are still people. What about the object itself? Are you measuring what you think you're measuring, and is it consistent? So you can send what's called a phantom, and we did this. We sent an inanimate object that has different levels of water, and we sent it around the country to multiple different sites and imaged it in an MRI scanner. And what it showed was that actually the biomarkers, some of the biomarkers that we're generating actually are very consistent and have high correlation with each other. Okay, that's great, but what about the measurements with actual measures of age, GFR, and UACR? Early on, we're starting to see that there are some signals and going in the directions that we think. In green means that the values are in the direction that we expect, that worse oxygenation will associate with worse kidney function and more proteinuria. Red means it's going in the wrong direction. So we're seeing correlations uncovered that are going in the direction that we believe them to be. And when we grade the biopsies themselves, which is one of the core plans of this project, we also see that some of the biomarkers in the imaging are associating with core lesions that we know are bad for you and we find in the kidney. And so more to be done on this, but as we recruit more individuals, I think we'll have a more salient picture of how these biomarkers associate non-invasively with lesions in the kidney. How can we use it in clinical trials, and how is it being used? Well, we typically are doing it in trials where it takes a long time to develop an endpoint in chronic kidney disease, but maybe it could be used as an intermediate endpoint. And so in SGLT2 inhibitors, it's been done where you incorporate an MRI and look at the changes in perfusion after you've incorporated or embedded into a trial and put somebody on an SGLT2 inhibitor. These data show that after you start somebody on empagliflozin, the perfusion actually does go down in people after they've been initiated on therapy. GLP-1 receptor agonist is another area to test this in, and we're doing this now, and that individuals who are receiving semaglutide or Ozempic are also receiving kidney fMRI, and perhaps we can get more insights as to why these drugs are working in the kidney the way that they do. Pirfenidone is a treatment already used for idiopathic pulmonary fibrosis, and is thought to be potentially an antifibrotic agent. And so some of the data that we've generated before have now stimulated other investigators to incorporate functional MRI to look at fibrosis in the MRI fashion instead of doing kidney biopsies and testing its response to pirfenidone. This is an ongoing randomized trial looking at this where they incorporate 3 different measurements of MRI over time. And this can be used as a surrogate endpoint, hopefully for fibrosis. And 2 last slides about how it's being used, and we're doing this currently, is that in people who receive a solid organ transplant of some sort, they receive calcineurin inhibitors, which are known to potentially lead to some degree of toxicity to the kidney and potentially fibrosis, and test this hypothesis. So in liver transplant recipients, some of them may go on and develop chronic kidney disease. And so we're embedding functional MRI into this trial where they're weaning people off of calcineurin inhibitors, putting them on a different immunosuppressant to see if it changes their kidney function over time. But importantly, are we seeing changes in the kidney after you're seeing response to drug therapy? So to summarize, the MRI biomarkers may provide valuable pathophysiologic and histopathologic insights non-invasively, and you can perform them in single and multi-center longitudinal studies. We need more data from these observational studies that incorporate kidney fMRI, that are paired with biopsy data to make sure that we're measuring what we think we're measuring. And longer trials of longer duration, larger and longer ones should incorporate MRI biomarkers to test the changes in physiology in response to treatment. I'll just acknowledge a lot of our collaborators. You can imagine a study like this would require a lot of people, a lot of buy-in. I'm not a radiologist, and so we really need a lot of us to work together. I have to acknowledge our funding from the NIDDK and overall NIH. And again, on a personal note, thank you to all of you, especially the patients who serve as the inspiration for the work that we do. Thank you.
Thank you, Dr. Srivastava. We appreciate everything that you've done. Dr. Sharma, thank you.